All right, so today I'm going to talk a little bit about the Seurat object that we downloaded last time. So I made a project folder here for our RStudio. I called mine Cosmix, COSMX, and I have a few folders here. So I have data, RENV. I found out that using the R environment is really nice to get all your packages, make sure that the versions and everything are fixed for this particular project. So if you open up another project, we have conflicts, as you can see down here. I'm not able to get this last one installed, but I'm going to work on that, and I can share how I get that working. I noticed I've had version or issues with installation of this package and getting it to run on later versions of R. Let's see if this version is here. Yeah, so this particular version here is 4.1.2. Using 4.3, I was not able to run in situ type, but it was installing. For some reason here, it says that the R lang is not found, but it clearly is. So if we do R lang, like it is here. So I might need to dig into the source code of this package and figure out exactly why it's not working. Today it's not going to be that important. So situ type is what's used in order to phenotype these cells. And if we go over to their GitHub page here, I already did some debugging and stuff to find out maybe there was a particular version of Rlang that it wants, but it works. Z is no longer at Nanostring, I don't believe. Yang. I might have to send an email. I have some people I work with there that has helped us with some of the information of the project that we have. But if we go back here, if we look at what we had last time, so we downloaded the Seurat object here. It's 2.23 gigabytes, and there's 100 and, I don't remember. Maybe we can go here. There we go. There's 194,000 total cells. Last time we weren't able to find out how many samples there were, but we'll be able to do that this time. I can do an interactive visualization and download the data. I think at a later time we're going to have to go and download the basic data files. Somewhere there is information about where the actual cells are. So here we can see that this is the transcript map. If I'm not mistaken, it looks a little too dense to be cells. But so something like this. We get the boundary of our cells and we can identify which transcripts belong to that. Maybe it's in the Seurat object that we have. I'm not 100% sure. When we start exploring, then we'll be able to find out. And so if you guys are interested, I can share this. I just saved it into our program folder and installing packages. Since this is a new environment with the RENV activate, all of these packages are going to be particular to this project, and it's like starting over every single time you run this. So if you follow these instructions, you should be able to get at least to here. That's where I was on R 4.2.1. So let's go ahead and create a new script. Library, let's first call Seurat. And it'll load here. Tells us potentially we're running an old version. That's okay. Actually, let's go ahead and do some headers here. So we'll do libraries. There we go. And we'll move this guy down below. Along with Seurat, as usual, let's do tidyverse. Tidyverse. I've gotten a lot more used to tidyverse. I used to be a basic R guy, but now let's install tidyverse apparently. I think it installed everything that's in tidyverse, just not tidyverse in particular. So let's do install.packages tidyverse. And what tidyverse is it has all of these packages within it, right? So it's got, you can see broom, dplyr, dbplyr, all those good things. And the thing that we mostly will use is actually dplyr. There might be some column selection with tidy select, I believe. Hopefully this runs pretty quick. If not, I'll go ahead and skip forward. But as that's running, we can set up our next thing. So we'll do Control-Shift, and that's how you enter in these new sections. Let's go ahead and call this one Load Data. Cool. Let's go back here. We have our data. So this is a Seurat object that we had downloaded before. So what we can do is go, let's just call it object. I feel like that That should be easy enough to type and fast enough to keep working around. We can do read, RDS, and then we need to go into our data folder, and then we can tab, and it should autofill. All right, it's taking a little longer than I would like down here to install all these. I should be getting pretty close. Yeah, there's not that many. Let's go back and talk a little bit about our data. So as I said before, in this, we're going to have our transcripts. Tertiary analyses. So this might have UMAP already calculated. We can go ahead and calculate our own if we wish. It will have phenotypes, so maybe we won't need to worry about in situ type. The nice thing about what they do, or what they did for us anyway, was that they have the phenotypes of cells that you would expect, right? So like immune cells, tissue cells. This is the brain, so they should have some different brain cells in there. But they also put in, this might be healthy tissue, but they also put in some unknown clusters. So with the UMAP, you can imagine it looks like this. Yeah, so they have unknown here. So let's see if we can find that just on the whim. It looks like it might be in here. Potentially, it's kind of a hard color to see. But what we can do then is take this, compare it to all these other ones with some clustering functions, and then look at the human protein atlas for single cell data and see actually what cells are being regulated here compared to the other clusters. And it doesn't look like they have immune cells actually, which is kind of interesting. They have neurons. Maybe the brain isn't immune heavy. That's something that we can explore as well. But with the unknown, we can go through and look at things like the human protein atlas. It's not as automated, of course. You're going to have to manually do some of that stuff, but you'll be able to phenotype those kind of on your own. What else is about this data? So when they do the Cosmics SMI, they have some metrics that they make sure that their cells can pass in order for them to be in the final Seurat object. And what some of those metrics are things like cell volume or area. If the area is too big, the segmentation might have failed, and you'll get doublets. So we can see, for example, if these two cells were too close together or potentially overlapped, because remember this is a 3D, this is a 2D representation of the 3D cell, right? So this isn't a perfectly flat tissue that they scan. So there might be chances or scenarios where the cells are too overlapped, the nuclei are not able to distinguish the two cells apart, so then we end up with this really large cell. Let's see if we can find, so like here, this might be real, but as an example, say that there's some DAPI standing here and DAPI standing here, and they're not able to segment those two cells out. They'll throw this away because they are unsure whether or not it's really one cell or it's two cells. If you get the raw data, you can go back through and actually look at those and see, okay, this is really one cell. Another thing that they do is make sure that the cells have enough transcripts in them in order to be phenotyped. So if you only have one transcript of these 6,000 genes, just remember they have 6,000 different genes that they're using to characterize these cells, then you're not able to phenotype them. If you're not able to phenotype them, you're not really able to understand what the tissue has. In order to do that, 20 transcripts is what we had. I'm sure with this, they might be a little bit more since they do have 6,000 different transcripts that they're looking for. Another thing is that they have negative probes. Let's go ahead and see if we can load this or if it'll give us an error. It looks like it's going. Good deal. All right. So there's negative probes. And if you have too many negative probes that show up positive on your sample, then there might be too much noise to derive real information from it. So in that case, you would also throw those away. Something that their institute type software, our package will do is take in those negative probes for their cells and then use that in trying to determine whether or not they're a particular phenotype. I don't know what association there would be between more negative probes for a particular phenotype or not, but it takes all that into account. And potentially it's not even a particular phenotype, it's just using that to remove extra noise. These Seurat objects are really large. So the one that I'm working with for work, after we run everything, is actually like over 4 gigabytes. It takes a really long time to read in. So you want to make sure that when you do read it in, you have, before you clear your environment, you want to make sure that you really want to clear it because it does take a really long time to read it back in. Oh, I thought this computer would be a little bit quicker. I suppose it's got to decompress it. So the RDS is actually a compressed file format, but I'll cut and then come back when this is read into our R environment. All right, we're getting it loaded in there. You can see it actually expanded to 14 gigabytes. I don't know whether or not that's accurate or if that's just what my computer has allotted to this particular object. Absolutely massive. So let's go ahead and open it up and see what we have here. So in the metadata, we can see that 188,000 of their cells had passed. So the metadata contains, let's go ahead and take a look here. Metadata. Cool. Metadata equals object, and it's an S4 object. So when we're working with it, we actually have to use the @ symbol, and then we can pull out this data frame. So metadata is a data frame. And if we go ahead and look at it, we can see here we have all of the different metrics that go along with our samples. So the FOVs, which are fields of view. There should be some information as to whether or not they use multiple slides. So here we can see they used some different markers here. Height of the cell, width of the cell, and then area. So this is what I was talking about before. They'll use sometimes this to threshold out doublet cells. We can see our negative probe counts, which are going to tell us how much false positives we have. We have DAPI, so this is going to help us detect our cells. When we look at our cell ID, we can see that we have some information here. This last one is going to be our cell. This middle one is going to be our field of view. So if we go back over to a field of view, we can see this first couple here are 100. Let's see if I can sort by this. There you go. Come back over here, and then we should have something like a slide. So you can imagine, I think each one can have something like 20 samples on a slide. And it's a tissue microarray. We can see what they were sequencing here or imaging, since it's the spatial molecular imager or imaging. There's our cell ID, What else is here? That's kind of cool. So we can see locations of cells. So we can actually do an XY plot of our different cells, looking at our fields of view. So if we subset to 100 or 1, we'll be able to see all the cells that belong to that, and then we'll be able to plot them, their locations. I think we'll have to go to the next page to see phenotypes, if they have phenotypes assigned. I'm not seeing it, actually. The last columns. So we got niches here. I don't see the cell types, though, which is interesting. So yeah, this video is just exploring. You can see the medium RNA. I thought they had, or would have had, some phenotypes in here. All right, let's go back to our object and keep looking around. So if we go into reductions, what this is going to be is dimension reductions. So we have these 6,000 different transcripts that we're looking for, and we can use those to reduce the dimensionality down to 2 or 3, depending on however many we want. So if we look at the PCA, we can see that they used 50 dimensions for our different cells. If we go down to their UMAP, we can see their embeddings. We might have to run a UMAP ourselves, which is okay. It just takes some time. Pretty cool. What else do we have here? It's not important, I guess. Miscellaneous. Ooh, so this is what's really, really cool about this data type. So if you look at the transcript coordinates, we can see the slide, the field of view that the particular transcripts came from, the location of that on the field of view, which probe they were using. So these would probably be ones that we would remove for an analysis. We can see the cell that they were assigned to. Yeah, we can see that they're controls. But we have, what is this? 163 million transcripts identified in this experiment. So this is where the large amount of data, so this is 14 gigabytes, comes from. What else do we have? So if we look at the assays, sometimes they'll have in here something like the SCT transformed, which is a Seurat single cell transformation, I believe, which does a little bit better than just centralized on zero kind of a deal, or normalizing RNA. False codes, pretty cool. But let's look at this metadata. We're going to have to explore a little bit to find out what we're working with here. Explore data. Explore data. Cool. So let's look at the different fields of view that we have. So we can do unique, metadata, $FOV. If you run this, wow, they have lots of fields of view in here. That's pretty cool. Let's actually build this into a table. So let's do metadata, and this is where the tidyverse will come in. Let's make sure we have this one loaded. Yeah, just telling us that it was built under a newer version of R. Okay, so we want to select our field of view, and if we press tab, we can kind of scroll through here and see what else they have. But I want to see if they have slide number, and they should. I believe that we pulled that out before, or looked at it. We'll have to go back to metadata. It's not called slide. 200. I probably passed it. Well, let's go back here. So there's our field of view area, so on and so forth. So run name is probably slide. Let's see if we hover, does it tell us? It's just a character. Let's do that. Let's look at run name. Oh, we have slide ID numeric. That one works too. Either one. We can do slide ID numeric. And then what we want to do is do distinct. So what this is going to do is it's going to give us all the distinct combinations of field of view and slide ID. And let's actually save this as our slide FOV. All right, so we have 392 fields of view. Wow. So we can look at field of view 100 and ID of 1. So we'll look at this cell here, and we can see the combinations. It just pulls the first one out. So slide 1, FOV1 equals metadata, and we'll filter. So if you don't load tidyverse and you try to run filter, it's actually going to try to use the matrix filter. You can actually check here. Filter. I should come up with a couple different options here. Stats. It'll use the stats filter, and it doesn't work the same way. So what we want is our field of view equal to 1, and we want our slide ID numeric also equal to 1. And this should give us all the cells in the metadata that are belonging to that particular field of view. So here we go. Let's just do an XY plot of this. We'll do slide 1, FOV1, ggplot, and then geome point. We need to set our aesthetics, And what we want is our X and Y locations. Names are a little bit different than what I'm used to. So we got X equals X FOV pixels. So for the Cosmics data, at least the one that we have, I don't know if it's the same with this, but each pixel is 0.1. Let's write this down. Each pixel equals 0.18 microns. So this is very, very detailed. All right, let's keep going here. And we can do y, y equals y FOV pixel. And then to make it look pretty, let's add in our coordinate fixed. I believe this is going to be the one that gives us the right aspect ratio. We can zoom. If we shrink it down. Yeah, there we go. So these are the centroids of all of our cells. And if we had, I don't think we do, but if we had those cell segmentation boundaries, what we'd be able to do is actually look at where those are. We have transcript coordinates. I don't think we have the polygon vertices. But what we can do with those is actually draw around our cells the size of the cell or the shape of the cell, which is really, really cool. Yeah, I already looked at that one. Active. So those are all the cells. Let's look at assays here. RNA. Data. This just looks like our cells by transcripts. So our cell, so the transcripts, they're going to have the negative probes and everything in there as well, but the cells there. And I don't see, hey, there's our genes, the probes. I don't see our polygon information. So that might be something that we have to go back and download later, which is completely fine. But here we have our slide. Let's go ahead and copy that one. I'll copy. We'll just plot slide 1, FOV 1. And there we go. We can actually make the color as well. Let's see. They have niches already identified, which might be kind of interesting to look at. So that's just the... here we go. So they do have a bunch of unknowns. So let's actually color our data by that. And then we can do color equals RNA NB clusters refined. Cool. Let's zoom in again. So we can see that there's a lot of endothelial. There's some astrocytes in here. Green is LNH. Let's see. Not 100% sure what that is. I need to bring that window back. Oligodendrites, microglia, lots of cool stuff. All right, what else can we explore here? Let's look at niches too. Let's copy all of that back to our script. And then let's do just called niche. Slide 1, FOV 1. Spatial clustering assignments. Oh, I don't remember anymore. Clustering assignments. And there we go. There's our different niches. You can see niche 4s, mostly over here with some niche 2. niche 8. Something that is very common in the brain are these layers, right? So if we go back here, we can kind of see those layers as they come across. Interesting. Let's actually look at a single cell as well. So let's start a new line, and we'll call it Exploring Cell 1 of FOV1 on slide 1. Okay, so if we go back to the metadata, we're gonna have to kind of, the object. We're gonna need to sort of work through this. Oh, it looks like we'll have to explore what we have for the cell IDs. So if we do object, and remembering it's an S4 object, we have to use @ then we can go into the miscellaneous, $, since that's on S4, it's just a list object. We can look at the transcript coordinates, and then what we want to do is we want to filter slide, lowercase, slide ID equals 1, FOV equals 1, and then what we can do is pull cell, let's go back. Gives me an error, that's fine. We can pull our cell ID of, we'll just pull cell ID, and then we can do unique, and what we want to do is we want to sort it so it's in order, and then we can do heads because we don't need to look at everything. FOV, lowercase. So lowercase FOV. Ooh, big data. Okay, so we can't actually look at slide 1 or cell 1, so we'll have to change that. But we can look at cell number 2, and then we'll be able to explore the locations of the cells or the phenotypes, transcripts in that cell. So let's do slide 1, FOV1, and then cell 2. Look at cell 2 equals, and then we can just copy this down here. Copy. Let's see if it worked. Ooh. And we can remove all of this. So what we'll do is we'll go into our transcripts, we'll filter into slide 1, FOV1, and then we can actually now add cell ID equals 2. Let's go ahead and run that and see what we get. There should be a lot of transcripts that are associated with this. Yeah, so this cell has 192 transcripts. We can see the different genes here. And then the negative probe. So let's go ahead and plot these. Plotting cell 2 transcript locations. All right, so slide 1, FOV1, cell 2, ggplot, geome point, AES, and we need to look at the column names for this. Column names of slide 1, FOV1, cell 2. Okay, just copy them up. X equals X FOV pixels, and then Y equals Y FOV pixels. There we go. There are the transcripts from our cell. Let's see if we can add a color. Equals target. So that's going to be the gene that we're actually interested in. Plus chord fix. And then I don't remember off the top of my head how to get rid of a legend. So we're just going to keep the legend and ignore it, because because there's a lot of targets. Ooh, all right. Give me a sec. We want to remove a legend. Ah, there we go. It's in the theme, plus theme. And then we want to do legend position equals none. There we go. So now we have the pixels in the right, the y pixel and the x pixel size is the same now. So this should be the real shape of the cell. And then we have all of the transcripts locations within the cell. If I had to guess, the nucleus would be here, in the area where there's big empty space. But we have the location of our transcripts in our cell. So I think that should do it for today. I know this video is getting pretty long. It's over 30 minutes. But what we did today was we explored the Seurat object that we have. We were able to look at the cells on an FOV and where they were located. And then we were also able to subset our a cell, subset down to a cell, and look at the transcripts that were identified in that cell in their locations, as we can see here. So stay tuned. Next time we will work on some PCAs and the UMAP. We'll rerun our own UMAP with the scaled. Let's go back to our object, the RNA scaled, normalized, and be able to see kind of where our different neighborhoods of cells are located. And it should look something like this for all of our FOVs. So yeah, thanks guys.